next session, session 1, which is called Genetics. By the way, I too would just like to thank Candace Utenza Hamilton for all of her support in bringing us together today, and Katja and the wonderful group at Stanford, because I think this is really an important endeavor and really great for all of those interested in 22q11.2 deletion syndrome. So without further ado, I'd like to introduce my very good friend and close colleague and collaborator, Bernice Morrow. Bernice is a professor of genetics, obstetrics and gynecology, and pediatric cardiology at the Albert Einstein College of Medicine in New York. Bernice has been instrumental in bringing the international community together. She was a leader from the genomics perspective for the International 22q11.2 Brain and Behavior Consortium, and she continues to lead in all areas as a trustee for the 22q11.2 Society. And Bernice is going to tell you about her work in congenital heart disease. She also has interest in hearing loss and brain and behavior, et cetera. But her talk today is entitled Human and Mouse Genetic Approaches to Identify Modifiers of Congenital Heart Disease in 22q11.2 Deletion Syndrome. And Bernice, the floor is yours.
Okay, can you see the screen?
Yes.
Okay, great.
You may want to make it full screen though.
Okay. Good?
That's perfect. Thanks.
Perfect.
Okay, okay. All right, so actually this is a beautiful picture of a mouse embryo done by one of the members of the laboratory, reminding you how amazing and beautiful mouse embryogenesis or mammalian embryogenesis is. So in our lab, we're interested in the genetics of 22q11.2 deletion syndrome, which I'll refer to it as 22q for time constraints. And our primary goal is to understand why some individuals with this condition have severe cardiac defects while others have no cardiac defects. And so we use human genetics approaches to identify genetic modifiers, and we also use mouse genetic approaches to understand gene function. So congenital heart disease occurs relatively commonly in the general population, but it's still rare at 0.8%, but it actually occurs in 60% of our cohort of individuals with 22q. And what's primarily affected in individuals with this syndrome is the cardiac outflow tract, which is shown here, which is the aorta, which pumps oxygenated blood from the heart and the pulmonary arteries. This region here is also called the conotruncal region in embryogenesis, and clinicians tend to refer to these type of defects as conotruncal defects, and the embryologists in the world call it outflow tract defects. And tetralogy Fallot is one example. In this case, you have malalignment, and instead of having nice oxygenated blood in red, you have a mixture of oxygenated and deoxygenated blood to the body, and this requires surgery in infancy for survival. So there are many deletion sizes in association with this syndrome. So as I mentioned before, congenital heart disease occurs in about 60% of individuals with typical features of the syndrome. And this shows the centromere and the low copy repeats A, B, and C, and D that cause the meiotic error leading to the deletion. And individuals have an A to D, A to C, or A to B deletion, but the most common is the A to D deletion, which is 3 million base pairs, in which you have half the number of the genes in the region as in normal, and the frequency is over 90%. So individuals with all of these 3 deletions have actually the same risk for congenital heart defects, meaning that critical genes for this syndrome are in this region. However, using clinical arrays in individuals with structural birth defects, a new syndrome was identified with individuals with non-overlapping deletions between LCRB and D, or C and D, resulting in a newly recognized condition. But in this condition, instead of congenital heart disease occurring in 60 to 75%, let's say, in the general population, it occurs in 20 to 30%. So we became interested in looking at two genes in the region, one associated with the A to B deletion, and this is a gene called TBX1, which Pete mentioned, and this encodes a transcription factor that regulates expression of genes. There's another gene called crackle, which is in the B to D region, and this gene is sitting in the cytoplasm and responds to extracellular signals. And these two genes were shown many years ago to genetically interact in embryogenesis. In fact, when you knock out this gene or this gene in mouse models, you get congenital heart defects. So our main hypothesis has been that loss of one copy of TBX1 and or one copy of crackle is the first hit in the genome, and genetic modifiers either on the remaining allele or elsewhere in the genome alter the overall risk for the severity of congenital heart defects. So the person in my lab who's mainly working on this project is a very talented senior postdoctoral fellow named Yingji Zhao. And her first project was to analyze whole genome sequence that was obtained from the IBBC, which is the Brain and Behavior Consortium, to identify genetic variants in the remaining allele of chromosome 22q in risk to heart defects. Her second project is to identify rare genetic variations that act as modifiers elsewhere in the genome. So as her first project, she looked for rare variants in genes on the remaining allele of chromosome 22. So here you have the two centromeres, the chromosome 22 region, the LCRA and LCRD. Here's the deletion on one copy, and here's crackle near the LCRD on the other copy. And she identified in individuals with the typical A to D deletion, which was about 1,000 individuals, that there are common variants that may be important for the expression of this gene crackle to influence the risk for congenital heart disease. Now, crackle is expressed in all cells in the body and in embryos. And so the idea is that if you have common variants around crackle, it could influence its expression and then thereby increase risk to having congenital heart defects. And then her second project is really to look at rare genetic variations elsewhere in the genome. And so in order to do this, she decided to do a case control comparison. So what's shown in this aqua color are the number of controls that we had whole genome sequence for as part of the IBBC, and that was a total of 537 subjects. There were 456 subjects that had conotruncal heart defects or outflow tract heart defects. And so she first did a quality control where she started with 21 million variants identified from whole genome sequencing. And then among these, she took the rarest that are variants that occur in less than 1% and have criteria of being potentially damaging, and identified almost 59,000 putatively damaging rare variants. And the focus here is on coding variants that encode for proteins. And she identified loss of function variants, missense variants that change an amino acid, and splicing variants that could cause a frame shift. And from these analyses, she identified high confidence set of putative damaging DNA variants. And then she further filtered these to genes expressed in the mammalian embryonic heart from data obtained from Li Ji, who is a senior graduate student in the lab. And from 21 million variants, she ends up with 2,000 what we call mutations in 1,008 subjects. Then she did many different types of analyses, including overrepresentation analysis, which is a statistical method. And she also used this weighted gene set analysis. And the gene sets were identified from a paper published from the Pediatric Cardiac Genetics Consortium to identify genes that may serve as modifiers to conotruncal heart defects. So among the genes that were identified by these approaches, what came to the top were essential genes for viability and chromatin regulators. And so this is a picture that was obtained using this string software with the help of Dr. Julie Seacombe, who is a colleague in the department. And what's shown here is these circles represent proteins that are chromatin modifiers. These lines represent how they're related to each other. And these colors represent the type of chromatin modifiers that they are. And of interest, for example, CAT6A, and all of these were identified, sorry, I missed something. All of these identified in either 2 cases and no controls. So you had to have a variant in 2 unrelated individuals. And these genes were identified, and CAT6A is among them. And CAT6A is of interest because it was shown by another group several years ago that CAT6A and TBX1 genetically interact. So, you wonder like why a chromatin regulator is found. And it makes you think about TBX1 and how that gene functions as a transcription factor. So here's a slide of a cell, and the nucleus is within the cell. And the nucleus is important for transcribing genes. And so TBX1 is a transcription factor, which is important for modulating gene expression. This blue line shows DNA, and these circles represent nucleosomes. And all of these other proteins are involved in transcription regulation. And these are chromatin regulators. And so the idea is that TBX1 binds, but it has to bind to open chromatin. And in order for TBX1 to bind to open chromatin, you need chromatin modulators to open the chromatin. TBX1 can also bind to promoter regions, and this regulates expression of downstream genes. And so when you have changes of gene expression, you have changes in cell migration, cell fate like differentiation, and the output of signals. And then on the other hand, you have the input of signals coming from outside the cell saying, hey, it's time to differentiate to a hard cell. And Crackle is sitting kind of in the middle of the cytoplasm sensing those signals. And so Crackle could, through signal transduction through proteins, bypass the nucleus and regulate changes in cell behavior, or it could affect gene expression. And so I could kind of understand why chromatin modifiers or chromatin regulators can actually modify the phenotype because of the fact that TBX1 is on 22q11. So what are our next steps in the human genetics? So one is to really look at the variants we identified and confirm that these rare, most damaging variants are identified in 2 different softwares used to call the variants, to experimentally validate about 200 variants by Sanger sequencing, which is just to look at the variant itself in the sample. And then we're performing whole genome sequence on more samples in order to expand the population. So we're performing whole genome sequence on 900 more samples that were supported by the NIH. And we're collaborating with Dr. Gurr and the members of the G2MH to obtain whole genome sequence from additional individuals with 22q. So this is the human genetics side. And then we're also understanding gene function and how genes interact with TBX1, and then variant function in mice and zebrafish. And I just introduce you to Daniela Dishornik and Yingji because this forms the team focusing on the human genetics side. So loss of TBX1, as Pete mentioned, causes defects. It causes cardiac outflow tract septation defects. So what this simply means is just look at this picture here. In this picture, you see a separate aorta and pulmonary trunk in a mouse embryo heart. It's beautifully shown in green, which are neural crest cells that migrate in from pharyngeal arches, which is shown here. Well, when you knock out TBX1, what happens? What happens is that these pharyngeal arches here which provide cells to the outflow tract, they go away. And instead of having a separate aorta and pulmonary trunk and tons of neural crest cells, you have this persistent truncus arteriosus, which is a failed septation of the outflow tract. And you can see how few neural crest cells in here. I mean, you don't have to be a cardiologist to see this difference. So interestingly, and we've known this for like over 20 years, that TBX1 is not expressed in these neural crest cells that go to the outflow tract. So how could this be? Well, TBX1 is expressed in what's called the second heart field. It's also called the cardiopharyngeal mesoderm. And this is showing expression in blue in an early embryo of, let's say, first trimester development. And TBX1 is shown in pink, and neural crest cells are shown in green. And these cells are intimately adjacent to each other in cardiac development. So therefore, TBX1 is expressed in one population, and it has to interact with the adjacent neural crest cells right next to it in order for these neural crest cells to know what to do and to form the septation of the heart. And so this is a picture generated by a postdoc in the lab named Christopher De Bono, and it shows the same thing I just showed you before, but more specifically. So here's an embryo, here's a head, here's a tail, here's the heart, and neural crest cells migrate from the neural tube, shown in yellow, into the heart. But neural crest cells interact with the second heart field mesoderm, shown in blue, in order to have a normal length outflow tract and in order to have septation to form the aortopulmonary trunk. So the mesoderm, which expresses TBX1, is important to signal to neural crest cells to interact to form the cardiac outflow tract and aortic arch. And this is what's disrupted. So what is the function of TBX1? On a single cell level, Hiroko and Chris have worked together on trying to identify the function in the second heart field and neural crest cells. And so Hiroko recently identified a multi-lineage progenitor cell population that actually is coming from studies of early first trimester mouse embryos, taking out cells, running single cell RNA sequencing, and identifying these red cells, which then are very, very important in differentiating to cells that will contribute to the heart and craniofacial muscles. And this, Robert Kelly will be talking about a little bit more about these fate choices. And so she found that these multi-lineage progenitor cells, when you knock out TBX1, it affects the ability of these cells to differentiate. And this is just a lot of data that is shown, but the most important take-home message is that TBX1 is important for progression of such cells into forming these different, more differentiated populations. And so to really get more data, we ended up looking at TBX1 function to understand the mechanism. And Hiroko Nomura also did chip sequencing to identify direct targets of TBX1, shown here. And then she also looked at open chromatin using a technique called ATAC-seq. So at the end, she identified that TBX1 acts kind of as in the hierarchy of genes for cardiac development by regulating directly and indirectly other important transcription factors that are expressed in these MLP cells shown here in pink, and that it's important for promoting cell differentiation to the craniofacial muscles and the cardiac progenitor cells. And this is done in collaboration with members of our Ledoux consortium. And so Chris De Bono, on the other hand, is looking at cardiac neural crest cells, the other side, and he's trying to understand what happens in the second heart field that affects the cardiac neural crest cells. And so he's identifying first what really defines cardiac neural crest cells, because it just wasn't known. And so he used single cell RNA sequencing to define the yellow and green and pink different cell populations based upon gene expression that are important in forming the cardiac outflow tract. And this is wild type, and this shows different gene expressions in different colors. And in the TBX1 knockout, you can see a total disruption in these cell populations. So he is looking at that. And how much more time do I have?
1 minute, Bernice.
Awesome. Okay, so Liji is a senior graduate student in the lab, and I just want to give a shout out for her because she has a thesis defense in a month, and she's trying to finish up, and she's looking at crack and crackle function in cardiac neural crest cells as they enter the outflow tract, and shows when you knock out these important genes in neural crest cells, you get fewer neural crest cells going into the heart, which really explains how TBX1 and crackle kind of interact. And so in conclusion, we use human subjects to understand why heart defects occur. We're using mouse models and collaborating with our friends who study TBX1. We're doing single cell RNA sequencing to understand cell fate. And so these are our main conclusions and continue to work on this. I want to give another shout out for the Croatia meeting. If you go to the 22q11 Society page, you could see this, and the dates are like June 26th through the July 1st. Please go to this website, and I want to acknowledge the people in my lab. I didn't get to mention the work of Hansu because he doesn't work on 22q, but this is the members of the laboratory and former members Sylvia and Hiroko, my collaborators at Einstein, CHOP, UPenn, and the Leduc Network. And of course, the IBBC Consortium, Emory, and families with 22q. Thank you.